Pleased to invite Dr. Uday Saxena for his talk on omics technology in target selection and drug discovery process. I would like to take the pleasure of introducing him. Dr. Uday Saxena is currently a co-founder of the startup biotech company Reagene Innovations. He has held executive and leadership positions at Parker Davidson and Arbor Heterogenics in Atlanta as VP Drug Discovery, Dr. Reddy's Laboratory as Chief Scientific Officer, US and India, and Carius Therapeutics CEO. He has a PhD in biochemistry from Memorial University and postdoctoral training at Columbia University. He was associated with the team at Park Davis, Pfizer, that discovered Liptor and atorvastatin, the largest selling peak sales of $17 billion drug in the pharma business. He led teams that brought several drug candidates from an idea into the clinic. And we are glad to hear from you, sir. Over to you.
Thank you very much, Sonalika. I'm going to tell you about omics technologies and specifically their applications in target selection and really all of the drug discovery process. So briefly, I'll talk about, I think I should introduce drug discovery to this audience because I'm not so sure what your backgrounds are. So I think I'll go ahead and do a brief introduction on what it takes to discover a new medicine. Then I will plug into that the role of omics, and clearly omics have a huge role in discovery of new medicines or drugs. And specifically, I want to focus on proteomics and its role in drug discovery. Right off the bat, I can tell you that 90% of the drugs that we have today have a protein target. In other words, if we did not have proteins, we would not have had a drug. So that's how important proteins are in drug discovery. And as a result of that, proteomics has certainly assumed a very important role in discovery of new medicines. So this is to those of you who may not be aware, but discovery of a new drug is a very long process. And when I say very long, it takes anywhere from 12 to 15 years. So the number one step that you have to have, as we already talked about in drug discovery and development, is you have to select a target. So the target selection, the protein that you want to go after and its association with the disease has to be very robustly demonstrated and convincing. So just showing something in a petri dish and saying, I think this is going to work is simply not enough because you're setting yourself for a failure. So everything starts with selecting the right target. So how do omics come into play here? Before we had the tools of the omics, like the genomics, transcriptomics, proteomics, and metabolomics, it was a little bit of a hit and miss, but now with these very sophisticated tools, you can actually understand the disease better. I think in summary for this slide, what I want to say is there is not one step in the drug discovery process in which we do not use some form of omics or not. So omics have really, really become very important in our quest for new medicines and drugs. I also want to tell you that omics are helping us understanding. So one is designing vaccine, the other is designing drugs, selecting target. I mean, all across the value chain of drug discovery, they're playing a very big role. Now, the other big role that omics are playing is trying to better understand the mechanism by which the drug will work. Yeah, omics have rapidly become the heart of drug discovery. There's no question. Protein targets are the basis of 90+% of the drugs. As you can see, mutations in the protein targets can cause dramatic changes, escape mutations. And proteomics is pretty much useful in target selection, drug selection, mechanisms of action. So long story short, long live omics. Because of omics, I think the likelihood of success of drug discovery is going to dramatically increase. So thank you very much for your time.